All right, ladies and gentlemen, so welcome back. Last time we were talking about Hardy-Weinberg equilibrium as a tool to express how a population could not be evolving as a way to create a baseline for what it would look like when populations do evolve. So the question is, we talked about all of those 5 conditions of Hardy-Weinberg as a non-evolving population. So what makes evolution possible then? And today that's where we're going to pick up. So, two of the biggest mechanisms for evolution are this idea of mutation and sexual reproduction. So, when we talk about evolution and natural selection, all of this thrives on variation within the populations. There has to be differential reproductive success, and the reasoning for that differential success has to come down to their genetic makeup. So the way in which we produce variation and then keep variation going in a population really comes down to these ideas of mutation and sexual recombination. So let's take a few moments and just remind ourselves of what we're talking about when we talk about mutations. So a mutation, remember, is a change in the nucleotide sequence of DNA. Something has happened through the process of replicating a cell, replicating its genetic information that has caused a change in those A's, T's, C's, and G's. Somewhere in that sequence, that coding has been changed. Now, depending on what that change then produces at a protein level, can cause either completely new genes to arise or potentially just new variations of a particular trait to come about. Interesting thing is it's not always possible to predict how that altered DNA will occur, when it will occur, and what its effects will be. We can make some assumptions, but when we get to talking about protein synthesis, we'll talk about that progression of genetic code to messenger RNA to amino acid sequence to then ultimately the protein it produces. But then we have to look at how much of that gets produced and what does that mean in terms of its ability to continue to do its job as that protein in the cell. And so there's really a lot of steps along the way where effects are possible, or sometimes that change in DNA doesn't really have much of an effect at all. So most mutations will occur throughout an organism's lifetime and will happen in their somatic or their body cells. Most of us are sitting here right now with a bunch of mutations, and we're all functionally working just fine. Because your body either clears those mutations and they never result in anything, or they are mutations that don't ultimately alter the way that your body works. When we get mutations in body cells that do ultimately have an impact on the function of that cell and potentially the function of that individual as a whole, then we can start talking about things in humans like cancer and other disease processes. But if those mutations happen in those body cells and do cause enough of a change to that individual, it causes that individual to die, but that's where the mutation stops. Because remember, the only cells that end up being passed on from parent to offspring are the gametes. So in order for a mutation to have any effect on an evolutionary scale, that mutation has to start in a gamete that then gets fertilized, becomes an offspring, and then that offspring started off as that single fertilized zygote, goes through cell division, and creates all the cells in its body that now also contain that mutation. So let's back this up, right? So if a mutation happens in your body, let's just talk about you, and it happens in your skin cells, okay? Too much UV radiation, too much fun in the sun without sunscreen, right? And so you develop skin cancer. That skin cancer is going to be what affects you, But you're not going to pass on that genetic mutation for skin cancer to your children. If that mutation, however, were to occur in an egg cell, that egg cell then becomes fertilized with a sperm, becomes a zygote, goes through mitosis, becomes a baby. Then we can start talking about cancers that are inherited, things like breast cancer. So somewhere along the way, that breast cancer mutation happened in an individual's body, specifically in a gamete, whether it was an egg or a sperm. That gamete got fertilized, produced an offspring, and now that baby, every cell in that baby, now contains that genetic mutation. And so if that baby is able to survive and reproduce, it will continue to pass on that mutation. Keep in mind, too, that not every mutation we're talking about has to be cancer or some sort of disease. It can just be a change in eye color. Right? We believe blue eyes are a genetic mutation. It was a mutation that occurred somewhere along the way that altered the pigment in your eye, and here we are with blue eyes. All right, not necessarily a detrimental mutation. So how do we get these mutations in the first place? Well, they can cause changes on a multitude of levels. Most of the mutations we probably think about are things like point mutations, single letters in the code that change. So it's a single base in a gene that has been altered. If we're talking about a substitution mutation, then we're literally swapping one letter for another. Like the picture shows, the gene should have had an A, mutation occurred, and a C got swapped in instead. These can have a huge range of impacts on the organism. They can have a significant impact if that change ultimately causes a change in the amino acid sequence, and that the amino acid that got swapped in or got deleted, whatever the case might be, significantly alters the shape of the protein it produces. We can also have the opposite end of the spectrum in substitution mutations where it's virtually harmless to the point of maybe not even knowing it exists. And these are the types of mutations that most of us here are dealing with, and we utilize it as a way to forensically identify one person's DNA from another. And so they're just harmless. They're just point mutations that didn't ultimately change the protein. Maybe it didn't change the amino acid at all, right? There's redundancy in the genetic code. Or the change that it did cause was relatively innocuous. So it occurs maybe at the end of the protein sequence, and it really isn't hugely impactful to the way that that protein gets shaped, gets folded, and ultimately does its job. Again, we often think of mutations as harmful, but they can also have an adaptive impact. So if we're talking about an organism who traditionally has brown fur, suddenly gets a mutation to have really dark black fur, perhaps that helps that organism camouflage itself in its environment even better, and then in which case that can be an adaptive impact as opposed to a negative one. Point mutations can also be frame shift mutations, where I either add or remove bases from the sequence. If you remember anything about your protein coding, all of that DNA and messenger RNA as a result has to be read in 3 base shots. It's a triplet code. So every 3 bases is the code for a particular amino acid. So if I were to go in and add an extra base, that's going to shift that coding sequence all the way down to the end of the gene, which means more than likely every amino acid beyond that mutation gets changed. Removal would be the same thing. If I take alpha letter, it's going to shift everything in the opposite direction, but it's still going to change all of the amino acids down the line. These guys are more often harmful than anything else, because I'm changing huge chunks of the protein structure as opposed to potentially just one small region. And then we can talk chromosomal mutations, which are an even bigger scale. And I'm taking huge chunks of the chromosome and changing it. Again, these are almost always harmful because I'm altering huge levels of instructions. That being said, there are instances where these can be neutral, and sometimes they even can be beneficial, especially if they end up leaving the genes within that chromosome section intact. This is also one way that we end up with situations where I get multiple copies of the same gene, and that can cause that extra copy to take on a new role based on some additional mutations that may happen. So how do we get chromosomal mutations? Most of these are a byproduct of that crossing over process that happens during meiosis. If I talk deletion and gene duplication, these are often two sets of the same coin, opposite sets of the same coin, I should say, where when we go through that crossover event, right, we're mixing up those homologous chromosomes between the genes your parents gave you before you go make the gametes for your offspring. And one homologous chromosome didn't get enough of the information, and that's where you end up with that deletion once meiosis finishes. And the other homologous chromosome kept a little too much, and that's where the duplication can come from. Sometimes they can actually completely invert, so rather than being swapped in the right order, they get flipped, and that's where those inversions come from. Translocations are a really weird one. So again, going back to that homologous chromosome idea, you're only supposed to do crossover between homologous chromosomes. So we have 23 pairs of chromosomes, right? If we're talking humans. And so you should only be swapping information from chromosome 1 that your mom gave you to chromosome 1 that your dad gave you. Between chromosome 2 that your mom gave you, between chromosome 2 that your dad gave you, and so on and so forth. When we get a translocation, now something has occurred that non-homologous chromosomes end up matching up. So maybe you get chromosome 3 linking up with chromosome 8. That's not supposed to happen. They carry different genetic information, but they linked up, they still did the crossover event, and now I'm mixing up information that doesn't even belong there. And so when your cells go through meiosis and then try to build that next generation, it starts going and searching for information and doesn't know where to find it. Think of translocations as taking a book on, I don't know, the Civil War in the library and taking it off the shelf where it belonged and now sticking it in the middle of the fiction section. Good luck finding that book. It's going to take you forever. If you ever find it at all, because it's not where it's supposed to be. And that's kind of what's happening in these translocation mutations, is that we're moving information and your cells just don't know where to go looking for it because it's not where it's supposed to be accessed from. So how often do mutations occur? Well, in most organisms they're actually relatively low. On average, we get about one mutation in every 100,000 genes per generation. In most plants and animals, mutations aren't going to be the sole means of evolution. That being said, we look at things like viruses and microorganisms, and because they reproduce so rapidly, they're a little more subject to mutation, plus just the fact that they create offspring so much quicker. Any mutation that does occur can become relatively widespread if it's beneficial enough in their population relatively quickly. RNA genomes also have a much higher mutation rate than typical DNA genomes do. Remember, RNA is single-stranded, DNA is typically double-stranded, and so DNA's double-stranded structure in itself tends to be more stable, and also because it's double-stranded has that backup to proofread itself. That's a lot harder to do in those RNA genomes because those are typically single-stranded genomes. That being said, mutations account for very few of the evolutionary pathways that happen. They're a great way to get new variation into the population if and when they occur, but most of the genetic differences we see from generation to generation have more to do with sexual recombination than they do mutations, if we're talking about a sexually reproducing organism, right? So when we talk about sexual recombination, we're really just talking about taking two parents, taking 50% of their information, and then putting it together to recreate 100% of the information necessary to make the offspring. And the way that those genes get shuffled out during meiotic division to make those gametes, and then the way that that information interplays in the random egg and random sperm that happen to find each other, creates all of this huge recombination, huge shuffling of existing alleles in a population. And even if you're talking about the same two parents, every time they go through a round of producing offspring, each offspring isn't going to be genetically identical, even though they have the exact same parents. Think about you. If you've got any siblings, unless you're an identical twin, you are not a clone of your siblings, even though you have the same two biological parents. And that's because genes get split out. There's recombination that happens in the process of making those eggs and those sperm. And then the fertilization of an egg and a sperm is also random, even within those two parents for every child they create. And then we can start talking about multiple parents creating children. So having half siblings and things like that, we're mixing things up even more. So sexual recombination really is the major driving force for keeping variation and producing new variation in a population when we look at it at an entire organism level. Mutations are only going to potentially get us some new traits at a particular gene locus.